Hi everybody. We're going to talk about coronaviruses today for obvious reasons. I think this is on all of our minds right now. And next week we're going to resume the course as originally scheduled by talking about hydrothermal vents. But even though I have spent my career studying hydrothermal vents, that's what I do, it's my thing, it just didn't feel right to lecture about weird microbes at the bottom of the ocean today. And I've been doing a lot of reading about coronaviruses anyway, and I thought I would share some of what I have recently learned with you guys. So I'm going to do that by discussing this paper with you. I don't expect you to have already read the paper before watching this video. I'm going to introduce it to you, and then I want you to read it after this video. And after you've read the paper and after you watch this video, then there's going to be an assignment about it posted on Canvas just below where you're watching this video right now. All right, this paper is about the diversity of coronaviruses that the authors found in a population of bats in a cave in China. And one of their big conclusions is that the evolution of coronaviruses in bats and their jump from bats to humans involved a series of recombination events. And I thought that was kind of a cool connection to what we were just talking about in this class before spring break, which was the importance of recombination in the speciation of microbes. So if you remember from before spring break, I had presented these two different models for microbial speciation. The older one was this ecotype model where everything is explained by random mutations and then a series of selection events that select those mutations. And you get genome-wide selective sweeps that wipe out all of the standing variation, leaving one survivor, which goes on to populate all the descending variation. And this model was developed at a time when we thought that recombination rates, that is the exchange of genes between populations, is very low. And recently, with the aid of better genomic data, and especially population genomics and metagenomics, we now appreciate that recombination, that is the sharing of genes between microbes, is much more common and perhaps in fact, incredibly important to how microbes speciate and evolve. And so we talked about this paper that proposed a model that recombination is constant, is going on continually in a population, and when recombination diminishes for some reason, for example, if there's some physical barrier that separates two populations and doesn't allow them to share genes as much anymore, then they can go on their separate evolutionary paths. And if recombination gets sufficiently low, then they are now genetically isolated populations that can become separate species. So this model has gone from mutations being basically the only thing that matters, and recombination is insignificant. So now recombination is the thing that matters, and mutation is the thing that happens, but not the primary means by which populations diverge and speciate. So this happens not only in bacteria and archaea, but as we're going to see in this paper, recombination also happens in viruses, which is not something you learn in intro bio. You learn that microbes are asexual, and viruses, if you talk about them at all, they are you learn that they're not true living organisms and that they require hosts to reproduce and are certainly asexual, which all of those things are not necessarily wrong, but it leaves out the fact that recombination does happen in viruses. So let's talk about that some more. So in this paper, they were looking for SARS-like coronaviruses in bats. This paper was published in 2017, so obviously before the current outbreak. And the reason why they were doing this work is because the original SARS virus, SARS coronavirus 1, which had an outbreak in 2002 and 2003, The new coronavirus that we're dealing with now has been recently renamed SARS coronavirus 2, or sometimes it's shortened to just SARS-2. So we have SARS-1 and SARS-2. SARS-2 is the thing that we're concerned about now. These researchers were doing the study because they were following up on SARS-1, which was known to have originated from some kind of animal reservoir, but it wasn't clear exactly which animal. So for example, they talk about here, and by the way, I'm highlighting in text, the author's text in yellow, and then my explanations are in red. So originally it was thought that civets, which are a type of cat, I encourage you to Google it, they're unusual, were thought to be the origin of the first SARS virus. But thanks to the work of, I think these researchers primarily and others, they appreciated that that bat populations also have a big diversity of coronaviruses. So bats are a potential origin as well. And the specific hypothesis from SARS-1 is that the virus probably started in bats, was somehow transferred to civets, and then civets were an intermediate host that then brought them to humans. And I think the main idea there is because humans in China are hunting civets, so there was a natural mechanism for how they might have interacted. So anyway, they say that despite this evidence, it's still unclear exactly how the jump was made from bats to humans. And so they want to dig into the genetic details of the viruses and figure out how this happened. And some of the confusion is that the coronaviruses that they know about from viruses are clearly distinct from the human virus. So here is some confusing terminology. They refer to the human SARS coronavirus as SARS coronavirus, SARS-CoV, and they refer to the coronaviruses that they have found in bats as SARS-R coronavirus, which stands for SARS-related coronavirus, which I think is a bit of unfortunate terminology that is not clear. But SARS-CoV is the human virus, SARS-R-CoV is the bat virus. So they say that the bat viruses that they've found so far are clearly distinct from the human virus in the S gene, which is one of the key genes in the virus, and one or more of some of the other genes like gene 3 and gene 8. ORF stands for open reading frame, it's a fancy way of saying gene. Suggesting that these genes don't provide strong evidence that bats are the direct host, are the direct origin for the human virus. And so they want to look at these genes individually and together to figure out what's the story, where did these viruses come from? All right, so to do this, they did a 5-year longitudinal surveillance, which meant they kept going to the same bat cave in China for 5 years, collected a bunch of samples directly from the bats themselves as they're hanging out in their cave. And from those samples, they assembled 15 genomes, 11 from this study and 4 from a previous study, for a total of 15 complete genomes of bat SARS coronavirus. So here they say to understand the genetic diversity of these bat coronaviruses, the most variable region of the SARS genome, which is the S gene. The S gene is the one that encodes a surface protein that is thought to be involved in the virus's recognizing and infecting the human or bat host cells. This gene is highly variable, which is probably part of the reason that the virus is hard to treat. So they did a PCR test, and in this table 1, CoV positive means it was positive for a PCR test, and SARS-R-CoV means these they sequenced their PCR products to confirm that they are what they thought they were, and so 64 of the 84 positives had, in fact, the gene sequence that confirmed it was SARS coronavirus, and not, for example, some other kind of coronavirus. And in fact, there are lots of different kinds of coronaviruses. The SARS coronaviruses are just one subtype within the larger family. Okay, so that's just a kind of a side note on their PCR test. And here's one of the key figures. So this is figure 1. Here they are using the SARS coronavirus from the civet cats as a reference. So in these gray boxes are the different genes of the SARS coronavirus. Gene 1b, gene 1b, the S gene, gene 3, and gene 8. And these curves are their 15 different bat virus genomes that they are comparing to the civet reference genome. And just shows that the 15 bat virus genomes are very similar to each other and are roughly the same amount of similarity to the civet virus over this part of the genome. And then when it gets into this highly variable S gene, there are a lot of differences among the different bat viruses and how similar they are to the civet virus. In other words, some of these bat viruses are much more similar to the civet virus than others. These are very similar. The y-axis is percent similarity. These are very similar to the civet virus. These are not so similar to the civet virus. So this is what I just said, and the conclusion is that the human virus isn't shown here. This is the civet virus, but in the results they report that the overall similarity between the bat viruses and the human and civet coronavirus, the human and civet coronavirus are much more similar to each other than they are to the bat viruses, is very high, significantly higher than observed for bat coronaviruses that I've reported from other locations in China, meaning that the bat viruses that they found in this one cave in China are the most similar to the human virus. These are the closest viruses to the human virus that anyone has found so far. So potential candidates of the missing link from bats to humans. This is what I just said, of a lot of diversity in the S gene. And then they have three figures 2, 3, and 4, that are very detailed comparisons of all the different virus genomes, drilling all the way down to the level of individual DNA sequence variation. And what they're doing is looking for similarities between the human and the bat viruses to try to figure out which one of these viruses is the most similar to the human virus and might be a direct ancestor. So here they're talking about similarities between human and bat viruses, just focusing on that S gene. So here are the different genes, and they're looking at differences among the bat viruses. Here they're looking at the actual protein sequences encoded by the genes, comparing all the genomes to each other. Here they're focusing on just that gene 8, focusing on just gene 3. And the point that they're getting to in all this details that I'm not going to go into is that they get different answers depending on which gene they look at. If they look at gene 3, they get a different answer of which bat viruses are most similar to which human viruses. So all of these details kind of come to a head in figure 5, which is the one that I want to focus on. So in the text they say both similarity plot and boot scan analyses, doesn't matter what those mean. Important point is that these analyses revealed frequent recombination events among these bat coronavirus strains. It was suggested that WIV16, the closest progenitor of the human coronavirus known to date, so the bat virus that is the closest to the human virus so far, was likely to be a recombinant strain from 3 different bat coronaviruses harbored by bats in the same cave, namely WIV1 RS4231 and RS4081. So this is the conclusion they made from figure 5. And so let's look at figure 5 to see how they came up with that. So here they are doing a similar plot as we saw before. These gray bars are reminding us again the different genes in the coronavirus genome. Gene 1, gene S, gene 8. And in this graph up here, which is the one that I'm going to focus on, they're showing 3 different comparisons. Each of the three comparisons involves this WIV16, which again is the one that is thought to be the closest relative to the human virus. That's compared to these three other bat virus genomes. And those comparisons are plotted here in these three different colors. And once again, in this part of the genome, they're all kind of the same. You get pretty much the same answer, although it turns out that WIV16 is the closest. The green bar is the highest to 100% here to this 4081. So this 4081 is the closest match to WIV16, which is the closest match to humans at this part of the genome. When we get to this gene, the blue comparison to the RS4231 is now more similar to WIV16 than the green curve. And then when you get to this part of the genome, we have a third answer of which of these bat viruses is closest to the closest human progenitor. Now it's this black one called WIV1 that is most similar to WIV16. So what this means is that if you want to know which bat virus is most similar to this bat virus, which is the closest to humans, then you get different answers to that question depending on which gene you look at in the bat virus genomes. And what this graph is indicating, which I'm not going to get into because it's highly technical, is that an explanation for the odd result here is that there was recombination among these genomes. That is, if you imagine that WIV16 is the product or the offspring of taking this part of the 4081 genome and then crossing that with this part of the RS4231 genome, and then crossing that with this part of the WIV1 genome, then you can get the exact makeup of the resulting WIV16 genome. In other words, their proposed explanation is that the way you get this WIV16 genome is by mixing genes from three different parent viruses. And this can happen in RNA viruses. Coronavirus is a type of RNA virus, and RNA viruses in particular seem to be good at doing recombination, and I posted a review paper to the Canvas page if you want to read about the molecular biology of how recombination occurs in RNA viruses. But the idea is that if two different viruses, or maybe even three different viruses, although it probably occurs in two different stages, so let's say two different viruses infect exactly the same cell, such that the RNA genomes of two different viruses are literally physically inside the same host cell, so the same bat cell or the same human cell, and then their RNA sequences get mixed up together during the replication cycle, then you can have recombination events between different parts of the viral genomes that then get repackaged into offspring viral particles that are the product of essentially a type of molecular sex between the parent viruses. Instead of molecular sex, we say recombination, that is, genes were recombined together to form the virus offspring. Here they do so that's the explanation for this result. In this figure, they are doing the same analysis, except this time with the civet virus instead of the bat virus that is thought to be the progenitor of the human virus. So here they're testing this WIV16 as the possible origin of the human virus, and here they're testing the civet virus as the possible origin. The main point, though, is that they get the same answer, that the civet virus also appears to be the product of a recombination event or possibly multiple recombination events between parent bat viruses. That is, the virus that is infecting the civets is also a recombinant of multiple bat viruses. This paper has a lot of data in it, and there was a lot of work that went into it. For example, the whole section of this paper involves some cool lab experiments in which they infect a human cell line inside their lab, so not actual humans, but human cell lines, with their new bat viruses to confirm that these bat viruses have the potential to infect humans. And they do a variety of lab experiments to prove that. They also make a phylogenetic tree to confirm that the bat viruses from this one cave are the most similar to the human and civet viruses. That's what this tree is showing. And finally, we're in the discussion section. Their big conclusion is that these findings provide strong genetic evidence for the bat origin of the human coronavirus with regard to the S gene or gene 8. However, all of these bat coronaviruses were distinct from the human virus in at least one of the other genes, suggesting that none of them was the one single progenitor of the human virus. Instead, you had to mix multiple bat viruses together in order to get the human virus. Moreover, these different genes that are most similar to the human virus were detected in multiple caves. The bats in this one particular cave in this study seem to be the closest to the human virus, but bats in other caves in China also seem very similar to the human virus, suggesting that the multiple populations of bats in multiple caves around China are mixing these coronavirus genes together among all these different populations. And this one cave in particular that is the focus of this study seems to be like the New York City or the Tokyo of the bat world, where all of these viruses are being collected in one large, densely populated site, where there's a huge variety of these coronaviruses that are all being collected and mixed together. So it's kind of a breeding ground for new virus evolution. And again, that evolution is occurring primarily due to recombination and not so much due to just random mutations. Finally, they conclude, yes, I think this what I just said. They go into some detail about how these recombinations might have occurred. They note that the way the interaction between humans and bats or civets probably occurred is due to humans hunting bats and or civets, but that they just don't have enough information to say exactly how that happened, or even if it is true that civets were an intermediate host, or perhaps the virus populations of civets were simply mixing together and recombining with the bat population, and it was all just one big mixing pot, and it's not clear exactly what the transmission route was to humans. They also note that they found these coronaviruses in multiple bat species, or at least 2 different bat species, indicating that it can hop between multiple bat species as well as to civets and humans, which is not surprising. If it can jump to civets and humans, it can also jump between multiple bat species. Here they talk about the different mechanisms of the human cell infection. And finally, in this last paragraph, they do a really nice job of summarizing their key results, which is, as a whole, their study conclusively demonstrate that all building blocks of the pandemic human virus genome are present in the bat viruses from a single location in China, and their data show that frequent recombination events have happened among these coronaviruses in that same cave. And finally, they end with this rather ominous conclusion. In addition, we have also revealed that various bat coronaviruses capable of using human cells are still circulating among bats in this region. Thus, the risk of spillover into people and emergence of a disease similar to SARS is possible. This is particularly important given that the nearest village to the bat cave we surveyed is only a kilometer away, which indicates a potential risk of exposure to bats for the local residents. Thus, we propose that monitoring of bat SARS coronavirus evolution at this and other sites should continue, as well as examination of human behavioral risk for infection and serological surveys of people to determine if spillover is already occurring at these sites and to design intervention strategies to avoid future disease emergence. So in 2017, these authors were noting that all of the ingredients are here for another outbreak. It already occurred in 2002 and 2003. That's the reason why they're doing the study, and they were well aware that this could happen again. Okay, that is this paper. I hope you found that useful to have me go through a paper and kind of explain it to you. I'm not sure if we're going to do this for the rest of the semester, but let me know. Did you like it? Did you not? And look at the instructions for the assignment on Canvas. And please send me any questions via email or during the online office hours, the details of which I will post on Canvas.